Cell cycle and cell division. Are you aware that all organisms, even the largest, start their life from a single cell? You may wonder how a single cell then goes on to form such large organisms. Growth and reproduction are characteristics of cells, indeed of all living organisms. All cells reproduce by dividing into two, with each parental cell giving rise to two daughter cells each time they divide. These newly formed daughter cells can themselves grow and divide, giving rise to a new cell population that is formed by the growth and division of a single parental cell and its progeny. In other words, such cycles of growth and division allow a single cells to form a structure consisting of millions of cells. Cell cycle. Cell division is a very important process in all living organisms. During the division of the cell, DNA replication and cell growth also take place. All these processes, that is cell division, DNA replication and cell growth, hence have to take place in a coordinated way to ensure correct division and formation of progeny cells containing intact genomes. The sequence of events by which a cell duplicates its genome, synthesizes the other constituents of the cells and eventually divides into two daughter cells is termed as cell cycle. Although cell growth in terms of cytoplasmic increase is a continuous process, DNA synthesis occurs only during one specific stage in the cell cycle. The replicated chromosomes, DNA, are then distributed to daughter nuclei by a complex series of events during cell division. These events are themselves under genetic control. Phases of cell cycle. A typical eukaryotic cell cycle is illustrated by human cells in culture. These cells divide once in approximately every 24 hours. However, this duration of cell cycle can vary from organism to organism and also from cell type to cell type. Yeast, for example, can progress through the cell cycle in only 90 minutes. The cell cycle is divided into two basic phases: interphase, M phase, mitosis phase. The M phase represents the phase when the actual cell division or mitosis occurs, and the interphase represents the phase between two successive M phases. It is significant to note that in the 24-hour average duration of cell cycle of human cell division proper lasts for only about an hour. The interphase lasts for more than 95% of the duration of the cell cycle. The M phase starts with the nuclear division corresponding to the separation of daughter chromosomes, karyokinesis, and usually ends with the division of cytoplasm, cytokinesis. The interphase, though called the resting phase, it is the time during which the cell is preparing for division by undergoing both cell growth and DNA replication in an orderly manner. The interphase is divided into three further phases: G1 phase, gap 1, S phase, synthesis, G2 phase, gap 2. G1 phase corresponds to the interval between mitosis and initiation of DNA replication. During G1 phase, the cell is metabolically active and continuously grows but does not replicate its DNA. S or synthesis phase marks the period during which the DNA synthesis or replication takes place. During the time, the amount of DNA per cell doubles. If the initial amount of DNA is denoted as 2C, then it increases to 4C. However, there is no increase in the chromosome number if the cell had diploid or 2n number of chromosomes at G1. Even after S phase, the number of chromosomes remains the same, that is 2n. In animal cells during the S phase, DNA replication begins in the nucleus and the centriole duplicates in the cytoplasm. During the G2 phase, proteins are synthesized in preparation for mitosis while cell growth continues. Some cells in the adult animals do not appear to exhibit division, example heart cells, and many other cells divide only occasionally, as needed to replace cells that have been lost because of injury or cell death. These cells do not divide further exit G1 phase to enter the inactive stage called quiescent stage G0 of the cell cycle. Cells in this stage remain metabolically active but no longer proliferate unless called on to do so, depending on the requirement of the organisms. In animals, mitotic cell division is only seen in the diploid somatic cells. However, there are few exceptions to this where haploid cells divide by mitosis. For example, male honeybees. Against this, the plants can show mitotic division in both haploid and diploid cells. From your recollection of examples of alternation of generation in plants, chapter 3, identify plant species and stages at which mitosis is seen in haploid cell.